Historic lows in the Chesapeake Bay's annual Blue Crab Winter Dredge Survey. The latest results show the lowest number of male blue crabs and the second lowest number of total blue crabs.
A river once filled with fish had become a silent graveyard, poisoned by pollution and starved of oxygen. By 2012, scientists were running out of options, so they took a risky step, releasing 10,000 juvenile blue crabs into the toxic water. Many expected the experiment to fail, but what happened next stunned researchers. The crabs survived, adapted, and began triggering a chain reaction that helped bring life back to the ecosystem. Could creatures this small really help reverse an environmental disaster?
Blue crab population in the Chesapeake Bay is on the decline. The news comes after 2 years of growth. A panel of scientific advisors said while the adult female crab population is up,
their story reveals how nature's resilience can shock even the experts. Crisis in the Chesapeake Bay. The Chesapeake Bay, once a thriving aquatic powerhouse, found itself spiraling into ecological collapse by the early 2000s. This vast body of water, larger than the entire state of Delaware, and a vital source of food and livelihood for millions, had become what scientists increasingly called a dead zone. Fish and other marine life struggled to survive in waters so depleted of oxygen that many literally swam to their deaths elsewhere. The riverbed was more graveyard than habitat, coated in organic sludge and choked by algae blooms suffocating everything beneath. This dire transformation did not occur overnight. The blue crab population, a signature species that once thrived in the bay, had crashed by over 70%. These crabs had long been apex scavengers, the janitors of the bay's underwater world, eating decomposing matter and recycling nutrients. Their decline disrupted the natural balance, allowing decay and pollution to run unchecked. As their numbers dwindled, the entire ecosystem edged closer to irreversible damage. For generations, the Chesapeake Bay supplied staggering amounts of seafood. At its peak, watermen harvested about 20 million bushels of oysters annually, enough to fill the United States Capitol building twice over. Oysters acted as natural water filters, cleansing the bay by removing algae and pollutants. But habitat destruction and overharvesting decimated oyster beds, stripping away nearly all of these natural filters. Without their cleaning power, the waters grew murkier, and the delicate food web began to unravel. The bay's crisis became a visceral example of how human activities can devastate an ecosystem. As fish disappeared and layers of algae thickened, oxygen levels plummeted to less than 2 milligrams per liter in large zones, where at least 3 milligrams are needed for most aquatic life to survive comfortably. Crabs required even higher oxygen concentrations, so these dead zones became inhospitable. The entire living system was collapsing into silence and stillness. These dead zones appeared almost predictably every summer across hundreds of square miles, smothering miles of water with lifelessness. What had once been a vibrant seascape buzzed only with the absence of sound and life. Scientists, fishermen, and environmental advocates watched helplessly as an area crucial for biodiversity and economic activity slowly suffocated. The crisis called for urgent and drastic intervention, but where to start remained unclear. The amount of pollution driving this collapse was honestly pretty staggering. Over 150 rivers and streams from 6 states funneled nitrogen, phosphorus, sewage, and industrial waste right into the bay, like it was nothing. Fertilizer runoff by itself kicked in hard, because industrial farming grew explosively, starting in the 1950s. That shift changed the water chemistry in a big way. Only about half of the fertilizers that were applied actually got absorbed by plants. The rest drifted into rivers, basically acting like fuel for massive algae blooms. When those blooms finally died off, they sank and decomposed, sucking out oxygen from the water in a relentless loop. Then there was urban runoff and a bunch of other pollutants that stacked on top of it. Oils, chemicals, and sediment clouds blocked the sunlight that underwater grasses needed, the same grasses that served as shelter for young crabs and fish. As those aquatic plants started disappearing, the juvenile crabs lost their nursery area, which made survival even harder. So the bay ended up stuck in this nasty cycle. Poor water quality caused species to decline, and then that decline made pollution worse, while habitat loss kept piling up too. All the warning signs kind of pointed to the need for an intervention, but the usual approaches seemed slow and also sort of limited by how huge the damage already was. The whole ecosystem had slid into this downward spiral, and honestly, nature by itself could not just reverse it. The disappearance of key species, the steady buildup of pollution, plus the lack of natural filtration, had left everything stuck in this deadlocked kind of state. It was more than an environmental issue, more like a breakdown of a crucial, interlinked web of life. While scientists were dealing with this catastrophic scene, one question hovered there. Could there be some way to kickstart the bay's recovery without waiting decades for slow cleanup measures to finally start showing results? The answer didn't live in machines or in chemical fixes. It was in the resilience of one single species. That realization basically set up a bold, unexpected route, the kind that would turn the situation upside down completely. The forces behind Chesapeake Bay's crisis laid bare a tough truth about human impact, but also hinted at nature's hidden capacity to rebound under the right conditions. Causes of the dead zone. At the heart of the Chesapeake Bay dead zone lies a perfect storm of pollution sourced from the surrounding mid-Atlantic region. Over 150 rivers and streams funnel water from New York, Pennsylvania, Delaware, Maryland, Virginia, and West Virginia into the bay, carrying an enormous load of agricultural runoff, industrial waste, and urban pollutants. Every inch of farmland, every factory release, every suburban lawn drenched in fertilizer feeds into the system like a vast funnel concentrating the problem. The industrial farming boom since the 1950s has been a primary driver. Unlike small family plots, these large-scale operations pump millions of pounds of nitrogen and phosphorus into the soil annually. Yet plants can only absorb about half of this fertilizer, meaning the excess easily washes into waterways. These nutrients act like a super fuel for algae growth once they reach the bay, sparking massive blooms that blanket the water surface. While algae might seem harmless or even beneficial at first glance, they are, after all, part of the natural aquatic food web. The problem arises when blooms grow too dense and eventually die off. When these huge algal mats sink and decompose, the process consumes oxygen at dangerous rates. This depletion crashes oxygen levels in the water to a point where few organisms can survive. The bay's infamous dead zones emerge every summer like clockwork, stretching for miles with near-zero oxygen below. But nutrient pollution wasn't the only culprit. The bay also suffers from a heavy load of urban and industrial waste. Sewage treatment plants discharge additional contaminants, while stormwater runoff from roads and cities carries oils, heavy metals, and sediment. The sediment inflow clouds the water, blocking sunlight vital for underwater grasses that provide shelter and food for juvenile marine species. These grasses are crucial nurseries. When sediment chokes them, young crabs and fish lose their habitat. This sediment clouding also makes the oxygen situation worse, kind of really worse. With no sunlight coming in to power photosynthesis, plants cannot bring oxygen levels back up, at least not in time. And when the underwater grasses start to thin out, the ecosystem loses a sort of natural protection, not fully obvious until it's already happening. The whole place slides further into decline, and the water becomes less friendly to most of the bay's usual life, as it breaks important life stages apart. Then there was the really catastrophic drop in oyster numbers. Before all this, people were pulling in something around 20 million bushels a year. Oysters worked as natural filters, pulling out algae and helping keep the water clearer. But between overharvesting, disease, and the loss of habitat, oysters got crushed, and their numbers fell by 99%. Once those filtration systems were gone, algae blooms started getting stronger and more frequent, and the dead zones kept spreading. After that, the blue crabs were basically next. They're aggressive scavengers, but also apex predators on the bay floor. When underwater grass is degraded and oxygen drops too low, safe nursery areas shrink. Then the poor water quality also messes with how well female crabs can produce eggs. So the entire life cycle begins to unravel, like a chain reaction, and you can see the breakdown of a system that once was thriving with activity. This pollution-driven spiral was not only an environmental disaster, but it was also economic and cultural, the kind that sticks around. For a long time, the bay was called a great protein factory, supporting fisheries, watermen, and local neighborhoods for centuries. When it declines, food supplies get squeezed, livelihoods get hit, and the region's ecological identity takes a lasting blow. Figuring out all these linked causes was crucial because it shows why the usual cleanup efforts were never going to be enough by themselves. The bay needed an answer that goes beyond surface fixes and actually brings back the ecological jobs that pollution erased. With the scope and complexity of the pollution clear, scientists began to reconsider the role of one species that might hold the key to the bay's recovery: blue crabs as a keystone species. Blue crabs hold a uniquely vital place in the Chesapeake Bay ecosystem. Despite not being the largest or most numerous species, ecologists classify them as a keystone species, one that plays a disproportionately significant role in maintaining the structure and health of their environment. Blue crabs act as janitors, security guards, and maintenance crews all in one, helping keep the underwater world balanced and thriving. As scavengers, blue crabs consume dead fish, decaying plants, and other organic debris that would otherwise fuel further algae blooms. By recycling these nutrients, they help reduce the amount of decomposing matter and prevent oxygen depletion in the water. Their feeding habits physically stir up the sediment on the bay floor, a process which aerates the soil and allows fresh oxygen to penetrate, vital for supporting various forms of life. They also control populations of species that might otherwise explode unchecked. For example, blue crabs prey on smaller fish and shellfish, preventing any single group from overwhelming the ecosystem. Their presence influences the behavior of other creatures, restoring a natural balance that supports biodiversity. Without them, delicate food webs risk crumbling under pressure from invasive species or overpopulation in certain groups. However, the health of blue crab populations directly reflects the condition of their habitat. The loss of underwater grasses, poor water oxygenation, and increased pollution have severely compromised their breeding and nursery grounds. Female crabs have produced fewer eggs, and hatchlings struggle to survive, leading to a dramatic population decline. This cascading effect threatens the entire bay ecosystem, since losing blue crabs means losing a critical natural regulator. Dr. Anson Hines of the Smithsonian Environmental Research Center saw this crisis and realized that blue crabs might be the ultimate solution. His counterintuitive idea: instead of waiting to clean the water before restoring crab populations, why not flood the bay with juvenile crabs to help the system heal? This approach was bold, risky, and contrary to traditional restoration logic. The crabs selected for release weren't ordinary wild ones. They were lab-reared juveniles, raised in hatcheries, fed optimal diets, and protected from predators until reaching a stronger developmental stage. These super soldiers had higher chances of survival than typical wild crabs, enabling them to face the toxic conditions in the bay more robustly. By reintroducing these keystone players, scientists hoped to restart the natural processes that had stalled, a biological reboot that could rebuild the bay's resilience from within. The crabs would not just survive but actively combat pollution and resuscitate the food web. It was an experiment in trusting nature's own engineered solutions. This plan challenged long-standing assumptions about ecosystem restoration and set the stage for a recovery story that continues to inspire new strategies globally. The outcome would prove whether nature's maintenance crew could bring life back to a place written off as lost. The reintroduction of blue crabs was more than a conservation effort. It was a test of nature's incredible capacity to heal if given the right support. The delicate balance of Chesapeake Bay's deterioration and the critical role of blue crabs set the stage for a daring intervention that might just turn the tide. The bold release plan. Faced with a seemingly hopeless battle to restore the Chesapeake Bay, a bold and unconventional idea emerged: Instead of waiting to clean the polluted waters before reintroducing key species, Dr. Anson Hines and his team proposed turning that logic on its head. They decided to release thousands of juvenile blue crabs directly into one of the bay's most damaged areas, betting that these creatures could jumpstart the ecosystem's revival. This plan was seen by many as reckless, even cruel, since the water was still highly toxic and oxygen-starved. The crabs chosen for this mission were far from ordinary. Raised in controlled hatcheries, they were provided with perfect diets and protection from predators during their most vulnerable early life stages. By the time of release, they had grown into late juvenile, nearly adult, blue crabs, robust, ready to face the harsh conditions they would encounter. The researchers hoped their advanced development would give them a fighting chance in the polluted environment. The Lafayette River, a small tributary on the Virginia side of the Chesapeake Bay, was selected as the testing ground. Though polluted, it was not entirely dead. It maintained some salinity and still held patches of underwater grasses, which could offer limited shelter for the crabs. These remaining refuges made it the perfect proving ground for the hatchery-raised crabs' survival ability. Timing. The release posed enormous logistical challenges. Releasing the crabs too early would leave them vulnerable to predators, while waiting too long risked colder water temperatures harmful to their survival. Coordinating transport, keeping the crabs stress-free, and releasing all 10,000 simultaneously required precision and careful planning. Each crab was measured and tagged with microscopic markers that would allow scientists to distinguish released individuals from wild populations later on. The tagging process wasn't just some scientific formality, it was quietly huge for figuring out whether the whole experiment would actually work. The team needed evidence that was clear and verifiable, so you can't wiggle out of it later. Not just maybe, but proof that the crabs survived and then started reproducing out there in the wild. In the end, that information would decide if the risky endeavor was mostly wasted effort or if it could be something far more transformative for the bay restoration. Lots of critics at the start basically scoffed at the plan. They said it was a doomed mission, a clear misuse of resources, and honestly, even cruel to the crabs. They claimed that it amounted to sending them to their deaths in a toxic place. That skepticism arrived fast and loud because this approach was so novel and bold compared to the usual rules, the established norms of wildlife management and ecosystem restoration. But Dr. Hines and his group still trusted the resilience and stubborn endurance of blue crabs. They're known for aggressive survival instincts, tough as anything, and that advantage might let them handle conditions that seemed impossible on paper. The idea was to shift the outcome using these super soldiers. Like technology alone hadn't been able to kickstart the process, at least not the way they needed. On a humid morning in June 2012, the real moment finally showed up. Researchers loaded crates holding 10,000 juvenile crabs onto boats and headed toward the murky waters of the Lafayette River. One by one, and also in tight bunches, the crabs were let go. Then they slipped away into dark green depths. After that came the worst part, the waiting. That first week felt too quiet, like the silence had weight. It was almost deafening. Would the crabs bolt immediately, or would they lose the fight to the toxic conditions? Nobody really knew. The river's secrets stayed hidden under the surface, and the fate of the experiment was balanced on a thin line, waiting to tip. Even with uncertainty looming, the team's gamble had set into motion a remarkable test of nature's will to survive. Early signs of survival. The first days after the release were filled with tension. Scientists placed crab pots in the river, unsure if the juvenile blue crabs had survived the toxic, oxygen-starved water. 3 days later, they pulled up the traps and saw snapping claws and scurrying shells. The tagged crabs were alive. Even more surprising, they were not weak or hiding. They were aggressive, active, and already claiming space in the polluted riverbed. Underwater cameras soon showed them defending territories, proving they were not just surviving, but adapting. Then came the real breakthrough. 3 months later, scientists discovered baby crabs that were not part of the original release. The females had mated successfully, and a new generation had survived in the same waters many believed were too damaged to support life. This changed everything. The crabs were no longer just an experiment. They were rebuilding a population and restarting the natural life cycle of the river. Sensors began showing small but important improvements. Oxygen levels near the riverbed started to rise, while layers of organic sludge began to shrink. The crabs were feeding on dead fish, rotting plants, algae clumps, and other debris that had been draining life from the water. As they moved, dug, and fed, they stirred the sediment and helped oxygen reach deeper into the riverbed. This messy natural process created better conditions for aquatic plants and other species. Within 6 months, underwater grass beds began recovering. These grasses provided shelter for young fish and crabs, creating a healthier cycle of life. More grass meant more oxygen, better habitat, and stronger support for the returning ecosystem. The river was no longer just a polluted wasteland. It was showing signs of renewal. The blue crabs had triggered a chain reaction, proving that one keystone species could help wake up an entire ecosystem. What began as a risky release had become a turning point. Scientists now had evidence that biological restoration could do more than save a few animals. It could help revive a dying river. From this growing success, scientists began to glimpse the broader potential for blue crabs to reshape more than just a single corner of the Chesapeake Bay, igniting hope for the entire dying estuary. Ecological domino effects. The resurgence of blue crabs in the Lafayette River sparked a surprising cascade of ecological changes that rippled through the entire system. Once the crabs began feeding and moving actively, they disturbed the sediment in ways that improved water quality and habitat conditions. Their burrowing and scavenging helped oxygenate the riverbed, breaking up compacted sludge that had long suffocated aquatic life. This natural tilling created an environment where underwater grasses, so critical to many species, could begin to regrow. Underwater grasses are more than just plants. They serve as essential nurseries and oxygen producers in the bay's fragile ecosystem. Before the crab release, these grass beds had plummeted to a mere 15% of their original coverage, choking off shelter and food sources for many aquatic animals. 6 months after the crabs arrived, grass coverage jumped to 23% and continued increasing to over 30% within a year. This bright green comeback provided refuge for juvenile fish and crabs alike, offering a foothold for broader ecosystem recovery. The crab's presence also rebalanced species interactions by suppressing populations of small fish and invertebrates that had overpopulated in the absence of predators. These smaller creatures had been consuming clams and other filter feeders that contribute to natural water purification. As the crabs restored predator pressure, clam populations rebounded, doubling the bay's natural filtration capacity. Clearer water allowed sunlight deeper into the river, further fueling grass growth and overall aquatic health. This intricate web of interdependencies kind of showed how the return of one keystone species can set off a trophic cascade, like a domino effect that ends up reshaping multiple rungs of the food chain. The crabs weren't just cleaning up debris, they were sort of directing the flow of life across the bay, often without any human guidance at all. Their aggressive feeding and burrowing behavior brought back ecological processes that had been resting in a dead zone, providing this new kind of blueprint for restoration plans, basically. And once habitat quality improved, plus food became more available, the bigger predators started to come back too. Rays, drumfish, and even smaller sharks began venturing into the Lafayette River, pulled in by the thriving blue crab population as a food source. Having those apex predators around is a pretty clear indicator of ecosystem health and its overall intricacy. It suggested that energy was moving upward through the food web again, restoring a system that could become balanced and self-sustaining. The river then shifted from a stagnant wasteland into this dynamic, living habitat buzzing with activity. Life at several trophic levels seemed like it was waking up, and it shows that carefully reintroducing key organisms can quickly recalibrate ecological balance. The crabs had become the generals of this biological renaissance, orchestrating recovery just by doing what they naturally do. Also, these shifts weren't limited to one place. They kinda hinted at how the method might be replicated in other areas of the Chesapeake Bay where ecosystems were similarly hurt. The cascading benefits from the crab release went beyond local gains, suggesting a scalable restoration model. The biological momentum triggered by a single species sparked real hope for one of America's most important estuaries, As this remarkable progress unfolded, it drew the attention of government agencies and conservation groups eager to expand the experiment. Program expansion and results. The undeniable success in the Lafayette River quickly inspired broader efforts to restore the Chesapeake Bay. In 2015, Virginia ramped up the program, releasing 50,000 more juvenile blue crabs across 5 additional sites. Each location mirrored the initial outcomes. Within 6 months, water quality improved, underwater grasses expanded, and fish populations rebounded. The results were consistent and measurable, turning a daring experiment into a proven method for ecological recovery. Maryland joined the initiative in 2016, further extending the program's reach. This bipartisan cooperation reflected growing confidence in the biological restoration approach, especially as positive data mounted. As word spread, the federal government began allocating funding, signaling institutional support after years of slow or ineffective traditional cleanup efforts. By 2020, over 500,000 juvenile crabs had been released throughout the bay's degraded areas. Scientific monitoring backed the observed improvements with hard data. Nitrogen and phosphorus concentrations declined notably in treated sites compared to untouched control areas. Fish diversity increased, signaling the return of balanced food webs and healthier ecosystems. These gains underscored how active wildlife restoration could accelerate recovery far beyond what passive protection or pollution reduction alone could achieve. Despite these encouraging trends, the Chesapeake Bay was far from fully healed. Dead zones persisted, and pollution, especially from agricultural runoff, remained an ongoing threat. No number of crab releases could completely offset industrial-scale nutrient input or the legacy of decades of environmental degradation. Yet the resilience shown by the bay's food web demonstrated that ecosystems possess remarkable self-healing capacities when the right triggers are in place. The crab release program challenged long-standing restoration paradigms that favored waiting for perfect conditions before intervening. It proved that even in less than ideal environments, keystone species reintroduction could jumpstart recovery processes. This paradigm shift influenced ecosystem management strategies worldwide, encouraging similar biological restoration projects in marine and freshwater systems. Programs in other regions took note. Efforts to restore kelp forests in California with sea otters and to repopulate coral reefs with specific fish species in the Gulf of Mexico mirrored the Chesapeake blueprint. The success of blue crab reintroduction became a case study in trusting nature's own machines to repair damaged ecosystems efficiently and sustainably. Despite lingering challenges, the program showed what happens when science embraces nature's complexity rather than attempting to control it with chemicals or technology alone. It opened new horizons for managing environments in crisis and gave renewed hope for some of the world's most threatened habitats. As momentum grew, so did broader conversations, not just about environmental restoration, but about our role in stewarding natural systems that are far more intricate and resilient than previously understood. With the Chesapeake Bay's recovery underway, the experiment with blue crabs illuminated new possibilities for ecological intervention, inspiring reflection on how humans might better collaborate with nature rather than fight against it. Lessons and future implications. The blue crab experiment in the Chesapeake Bay changed how scientists think about restoring damaged ecosystems. For years, the usual idea was to clean polluted water first, then bring wildlife back later. But releasing 10,000 juvenile blue crabs into a toxic river challenged that thinking. Instead of waiting for perfect conditions, researchers gave nature a chance to begin healing itself. The results showed that ecosystems can be stronger than expected. The recovery did not come from machines or chemicals, but from the natural behavior of a key species. Blue crabs helped clear debris, stir sediment, and reconnect parts of the food chain. Their survival proved that even polluted environments can recover when the right natural forces return. Still, the Chesapeake Bay is not fully healed. Pollution, dead zones, and runoff remain serious problems. But the experiment showed that recovery doesn't have to be perfect. It needs momentum. Once the crabs restarted some critical biological processes, things slowly started improving for other forms of life too. And the lesson goes way beyond just one river. All around the world, conservationists are thinking about restoration a little differently now. Rather than only leaning on technology, they are leaning toward nature's own repair rhythms, like reintroducing species that help maintain the balance of ecosystems. At the same time, this method also brings up big questions. Big, large-scale species reintroductions have to be handled carefully because ecosystems are complicated and not fully predictable. Helping nature recover doesn't really mean steering it like a machine, not completely anyway. In the end, the Blue Crab Project reminds us that humans aren't the only problem solvers. Nature already holds powerful tools for survival and renewal. Maybe our role is to support those tools, not take their place. The story of 10,000 crabs proves that even in damaged places, life can endure, rebuild, and, yes, bring hope back for the future. The remarkable revival sparked by releasing 10,000 juvenile blue crabs into a toxic river teaches us that nature often holds the keys to its own healing. This bold experiment changed restoration forever and proved the power of partnering with ecosystems rather than waiting or working against them. If you found this story inspiring, don't forget to like and subscribe for more incredible tales of nature's resilience. And before you go, tap the next video on your screen to discover another surprising story where nature fought back in a way no one expected.